Unit 1, topics 4, 5, and 6, biological macromolecules. We're going to talk about the properties, the structures, and functions. Nucleic acid. Technically, a nucleic acid is a polymer, and it's made up of monomers called nucleotides. The function is to store, transmit, and express information that is inherited. And it comes in these two forms, deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. In a hierarchy naming system here, the nucleotides are the smallest monomer. Many nucleotides ultimately put together in a sequence are going to create a nucleic acid. But when one nucleotide attaches to another nucleotide, we call that a polynucleotide. And many polynucleotides structured together are going to create the nucleic acid. So the nucleotides are made of 3 components. They're made up of a nitrogen base and a 5-carbon sugar. That 5-carbon sugar we call pentose, and then a phosphate group. So in a polynucleotide, each monomer only has one phosphate group. So if I were to draw that phosphate, which is PO4, has a bonded oxygen, oxygen, oxygen. This is our phosphate group, right? And it's bonded to our pentose sugar, which is a pentagon like that. There's like a CH2 here. And then off of our pentose sugar is the base. So sugar, phosphate, and the base. It's the difference in these sugars that give it a name, DNA or RNA. If it is a deoxyribose sugar, it gets the DNA part, and if it's a ribose sugar, it gets the RNA part. Pyrimidines and purines are 2 types of bases. So pyrimidines are 1 ring with the 6 atoms. So cysteine, thymine, and uracil are all pyrimidines, and the purines are adenine and guanine. And it's a little obvious maybe to tell that why they are in 2 different groups is because, one of them has 1 ring and the other one has 2 rings together. Oh, and so before I got ahead of myself. All right, so these 5-carbon sugars are bonded to the base. In DNA, we see deoxyribose. In RNA, we see this ribose sugar. And these sugars differ in structure, but they also differ in how they function. Can you spot the difference between the 2 sugars? So actually, the only difference is this oxygen right here. Deoxyribose lacks the extra oxygen atom on the 2nd carbon ring. So the phosphate group, or PO4, is added to the 5' carbon in the carbon sugar, and it's attracted to the base, and it forms this nucleotide. Why it's called 5' is not very well illustrated. Let me draw it. So here I have, right here, it's deoxyribose. Each of these little bends, like you see a little kink, like a kink right there, a kink, 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 represents a carbon. And the carbons are numbered based on IUPAC nomenclature and rules. So each little kink has a number. So this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So 5' is that carbon. And so this, if we were talking about this carbon, that would be carbon 1'. This would be, carbon 2', etc. Polynucleotides, these phosphate groups, they're linked adjacent to their nucleotides based on what base it is. What base gives it its directionality. It's called a phosphodiester linkage. So the directionality is that it's going to go from the 5' end to the 3' end. And the sequence of bases along the DNA or the mRNA is unique to our gene sequencing. but it will dictate the amino acid sequence ultimately in the end. And so, dictating this primary structure of a protein will ultimately dictate the 3D structure of a protein. DNA is going to consist of two polynucleotides. They form a double helix, and the strands are anti-parallel. They're held together by this hydrogen bond between the bases, and that's how we consider them bonded, all right? So cysteine bonds with guanine and adenine bonds with thymine. So you may remember that for every C, you're going to get a G, and for every A, you're going to get a T. In this case, though, we're looking at directionality. If you look in this figure over here, this is our adenine, and this is our cytosine, and this is our thymine, and this is our that's guanine right there. And so that you know that A should pair with T, right? But the directionality is how the sugar's pointing. So like when the pentagon is pointing this way, it's pointing in the 5' direction. If you look at the other side over here, the pentagon's pointing that way, and so it's starting at the 3' carbon and moving down towards the 5' carbon. And so strands of DNA are what we call anti-parallel. RNA is different not only because of the sugar, but it is also a single-stranded polynucleotide. It's going to vary in different sorts of shapes, and it also is going to vary in that base pair. So instead of thymine, we have uracil. All right, so if you were given a segment of DNA, would you be able to determine the complementary strand? If I'm starting with the 5' end going to the 3' end, C, A, T, G, can you come up with the opposite anti-parallel strand? Well, opposite will be the 3', and then followed by for every C, we're going to pair that with G, for every A, a T, for every T, an A, for every G, a C. So go ahead and do the rest of the strand, check your answer. Let's do some more practice.